कुछ प्रॉब्लम चल रहा है?
जी सर।
कुछ प्रॉब्लम हो रहा है?
नहीं सर। सर ये क्वेश्चन आंसर के लिए सर अलग से बना रहा था सर। आप जब क्वेश्चन आंसर करेंगे तो सर ये सीन क्रिएट किया है सर।
अच्छा,
इसमें तो सर पीपीटी रहेगा सर।
ये पीपीटी है?
हाँ सर। और सर जब क्वेश्चन आंसर के लिए सर ये सीन क्रिएट किया है सर आपके लिए सर।
कहाँ पे? कौन बोला? साइड में?
हाँ सर।
अच्छा ठीक है। अच्छा इसको डालेंगे अभी?
हाँ सर।
डाल देते हैं। वो स्लाइड को भी निकाल दीजिए हम लोग। स्क्रीन पे आ गया
सर। इसमें
सर।
सर पीपीटी होते ही सर एक बार सर इस पे आना सर, ओबीएस पे सर।
कहाँ पे?
इसमें आना सर, और सर सीन 2 यहाँ पे सर क्लिक कर देना सर।
ठीक है, इसे क्लिक करने से क्या होगा?
सर यहाँ क्लिक कर देंगे तो सर ये हमारा आएगा सर, ये व्यू आएगा।
हमारा जो कैमरा वाला भी है।
हाँ सर। और फिर सर इसके बाद जस्ट सर क्रोम पे सर जो क्वेश्चन आंसर के लिए सर।
ठीक है, और वैसा खाली पीपीटी दिखाना है तो
सर उसके लिए सर सीन 1 सर।
सीन 1, उसके बाद पीपीटी।
हाँ सर, पीपीटी सर। सर मैं इधर ही रहूँगा सर, थोड़ा
नहीं कोई बात नहीं, आप तो मैं पीपीटी में ये दिखाना है मोस्टली।
सर थोड़ा हमें मॉडिफाई करना चाहता हूँ सर, तो
ठीक है ठीक है, कोई बात नहीं। अटेंडेंस कितना आया है?
अभी कोई लाइन नहीं आया सर।
अभी कोई लाइव नहीं आया?
सर कुछ भेज दिया सर दूसरी।
कहाँ पे? यहाँ पे?
जी सर, सर समथिंग सर।
यहाँ पे?
जी सर।
अटेंडेंस कितना आया है?
आपकी क्लास है आज?
हाँ क्लास है।
ये कौन है, ऑडियंस कौन होगी?
और एमडी और एमएससी।
लेट आई आपकी इनकम।
हमने कल ईमेल कर दिया था।
संडे को कोई देखता नहीं ना।
अच्छा अच्छा अच्छा, ठीक है। हाँ मैंने रेसिडेंट को बोला था सैटरडे को, लेकिन शायद वो भूल गया था।
उनके भरोसे मत आता है, आप खुद कर दिया करें खुद।
हाँ ठीक है, आगे से मैं खुद ही कर दूंगा। थैंक यू। अभी एक जना है। अच्छा, कल परसों भी मेरा क्लासेस है, उसको 11 से 12, उसको भी बुक करना होगा।
जी सर।
ठीक है, ये सर इंचार्ज है?
सर।
अच्छा, क्या नाम इनका? ठीक है, मैं जाते वक्त तो रजिस्टर में लिखा के जाऊंगा। अच्छा, सीन 2 में ये दिखता है अपना कैमरा व्यू है। अच्छा, और सीन 1 में ये वाला व्यू। अच्छा, ठीक है। ये खाए वो जो सोनिका
सर वो अपने माइक सर ये वेबकैम ही सर
इसी में माइक्रोफोन है।
हाँ जी सर।
अच्छा ठीक
इंटरनल होता है
है।
सर।
अच्छा नहीं पहले था ना गोल जैसे क्या पे था।
सर उसमें थोड़ा ऑडियो इशू आ रहा था।
अच्छा
स्लो ऑडियो था सर उसमें।
तो इसमें भी ठीक है। ठीक। Yeah, good morning. We are going to start in next 2 minutes. Okay. Good morning. We are going to start in next 2 minutes. आपने पढ़ना है? पढ़ना है? ठीक है मैं चालू कर रहा हूँ. Okay, good morning. So we are kind of going for our first class on systems biology. Why I say first? We are going to have about 4 classes in this one. I'm trying to squeeze the 4 class into 2 class or the max 3 class because of the logistic issues of coming over here and then kind of booking this entire place. So seeing all those variabilities, I'm just trying to squeeze the 4 classes into either 3 class or 2 class. So let me know. I mean, your feedback. What do you think? Whether it is reachable to you, whether you could appreciate the class, or if you need more class, we can always do it. Okay. So let's see. So today is the first introductory class on systems biology. Now, before we begin this class, I just want to condition our mind in something like what exactly we do in systems biology. So for the biology, what we kind of been studying or we kind of discussed over here, including everything, whatever has happened, we try to understand the system, a complex system, by reducing it to simpler components. I mean, that is the conventional approach of biology. I have a few slides to show you that for the last 200 years or 300 years, the way in which you used to study biology is that you take a complex system and then reduce that complex system into simpler components and then study each of those components and go back and extrapolate and try to find out the whole about the system. I'll just give an example that if you want to study, say for instance, any cancer, say breast cancer or pancreatic cancer, a bladder cancer. So what you do is that you take a big chunk of the tissue from OT or from OPD or wherever it is, and then we are going to chop them into small pieces, right? And then take those small pieces and then isolate the DNA or RNA or proteins, whatever you want to, from those pieces, and then do a PCR or sequencing or Western blot or cytology of this one, or histology of those sections of those tissues, and then eventually you get some data, and from that data you try to go back and relate it to the actual original cancer. So you take a complex system, break down into smaller components, and then do the biology or the analysis or whatever you wanted to do on those small components, so those simpler components, and then correlate back to this complex system. Now this is what is known as the reductionist view of looking into biology, where we reduce a complex system into simpler components. There is absolutely fine in that one. There is no problem in this. But the problem comes when you have a complex systems like mammalian systems or in eukaryotic system where there are millions and billions of cells. Then the problem comes is that what the problem we encounter, that not every time if you reduce a complex system into smaller components, you can go back and then extrapolate it back to the original scenario. And that is the reason why many of our clinical trials fails, many of our drug discovery pipelines does not give us any novel drugs. Many of the experiment fails. The reason is not not the technicalities of the person who has been doing it, but rather the approach or the mindset in which we try to address the thing. And that is where the systems biology is going to be very important. So systems biology is looking into a complex system as a complex itself. So rather than breaking down into simpler components, you take a complex system and take into account that this system is complex and then do the biology rather than reducing it into simpler components. Because when you reduce it, then you cannot go back to the original system, and there comes the problem. So as and when we go through this PPT, it will become even more clear to you that why we need to study systems biology. But before we enter into the philosophy of this one, there are few things which all of us should appreciate, and that is every discipline or every domain or every subject has its own jargons, right? Like if you study biology, you need to study or put a few jargons, a few specific words for that particular system. If you study mathematics, you need to use certain mathematical terms. You cannot use any random words. Similarly, if you study chemistry, physics, law, whatever the subject you study, that particular subject has its own jargons. So systems biology has its own jargons, and we are going to see and learn those jargons. Why it's important? Because if you are going to start a, a class on systems biology, then and if you happen to see a book on systems biology, then unless and until you are going to get used to those jargons, it will be difficult for you to understand the whole subject. Okay, so bear with me, those are not very complicated. We are not studying hundreds and thousands of those new words. I'm just cherry picked a few of them, very few, the bare minimum which we should know. That's the bare minimum which every one of us should know in order to take systems biology as one of the curriculum in your syllabus. Okay, so from these introductions, let me go to the next slide. Okay, so that is the next slide. Hopefully the next slide is displayed to you right now by this time. Okay, let me see, because there is a little lag which is going on, so bear with me for the next slide. Okay, so the next slide is starting with what you call as what do you define a system. How do you define a system? So a system is a group of, so this is a very important definition, we cannot forget this definition. So what is a system? When you say systems biology, we need to understand what is a system in the first place, right? So a system is a group of interrelated, see there's a very confusing words, interrelated, interconnected, interdependent and interacting elements that forms a complex whole. What does that mean? That means when you are studying systems biology, then we are talking about stuffs or talking about things which are related with each other, so interrelated, which are connected with each other, that is interconnected. So there is a difference between relation and connection. When you say relation, it often means binary relation. So A is related to B and B is related to A. That's a relation. But when you say that A is related to B, A is related to C, A is related to D, A is related to G, then no longer it's a binary relation. Then it becomes what you call as a network relation, right? So then we use the word not interrelated, but we use the word interconnected. So systems biology means a system is actually a group of interrelated, interconnected. I told you the difference between relation, interrelated, and interconnected. Interdependent. What does the interdependent means? Means the two things are actually being joined together. So if you going to change one, it is going to affect the other one. So they are also dependent on each other. So interdependent and interacting systems. Why do we say interacting? Because the two systems actually are the two components of the systems. We are going to understand everything of them. Okay, once you bear with me, a few of the slides, you are going to understand everything of them. So what is interacting elements means each one of those components of the systems has an affinity for each others, so they can interact with each other, they can cross talk with each others. Just like when hormone binds to the receptor, we know that there is a hormone receptor complex, and then eventually that hormone receptor complex initiates a cascade of intracellular signaling, right? So this is an interacting systems wherein hormone bound to the receptor is actually interacting with the cell, and then the cell actually initiates its signaling cascades. So all of this together is what we study in systems biology. So systems is a group of interrelated, interconnected, interdependent, interacting elements that forms a complex system. So any complex systems will have all of these things which is going on in them. There is an interrelation, interconnection, interdependent, and interaction. Okay. And if you have again a difficulty in understanding, get back to me later after class that what exactly we mean by this. So just for the second time, I'm just trying to explain to you this these terms which are very confusing, but apparently they are very simple if you actually understand it properly. So interrelated means when there is a binary relationship between these two. Interconnected means when there is a network of relation. One thing is related to 100 different things. That is interconnected. Interdependent means when one is dependent on the other. That is known as interdependent because they have been joined together. So if one changes, the other changes. That's interdependent. And interacting means when the two systems or two components of the systems actually interacts with each other. Like I have given you an example of the hormone receptor complex. Okay, that's the thing. Now, the best example of systems biology is the human body. It's been regarded as a functional physiological unit, right? Now, just think about this. I mean, we have been teaching to the students that if there is a decrease in the blood volume, what happens? Or if there is an decrease in the blood pressure, what happens? How the system gets activated? How the suppose whenever there is a decrease in the blood volume, how the renin angiotensin systems gets activated, or how the vasopressin system gets activated. So that is a very complex system where there are a lot of feedback loops mechanisms which kind of tries to bring the system back to its original state if there isn't perturbations from that. So human body has been regarded as a very functional, complex physiological unit where systems biology plays a very important role in understanding these complex systems. You cannot use any other form of biology to understand an organism as complex as a human being, right? Because there are so many different interacting, interdependent, and interconnected components within that human complex system. The other examples are the society, which is again an example of the complex system. So these are the examples of the complex systems. Why is it society? Because in the society, when a society has been there, housing society, suppose, we have so many different things, right? The people living over there, the security guards, the facility managements, the people who manages the flowers and the horticulture departments. So you see there are different people with different speciality which actually chips in to maintain that housing complex society, right? AIMS society, think about it. So many departments are actually been integrating with each other to run a big complex system like the AIMS campus. So these are all the examples of a very complex systems. Now the Department of Biochemistry, which I actually mentioned at the end of that example, is another example of a complex interacting system. So we have this head of the Department of Biochemistry, and then under him we have the 16 different faculties, right? And each of the faculties has their own research labs. So they have been not connected just to the lab, they have also been connected to the departments, right? They are the department of teaching programs, mentoring students, and other things. So this is a very complex system. You could see that from one person, you have 16 different persons, and each of those 16 persons is been again associated with so many different activities. So this is an example of a complex tree or a complex system. And human system is another very complex system, right? So these are the systems where in order to understand them properly, we need to understand them in terms of systems biology. We cannot understand them with the help of conventional biology. How and why, we are going to see later as we are going to go into more of the different slides. Okay, so I'm now switching to the third slide. Okay, the third slide talks about what are the features of a system. So what do you mean by the features of the systems? So that's the third slide, which talks about the features of the systems. Now again over here, a few of the jargons which I was actually mentioned right in the beginning of the class, that we need to be a little accustomed with the jargons, because systems biology is itself is a known discipline. So we need to learn those terms. Not all of them, but a very few of them. Bare minimum one, but we need to learn them. Okay. Here are a few of them. These are a very, you know, probable exam questions. Certainly someone can ask you that what is the purpose, what is the environment. So I'm going to go through each one of them very slowly and trying to make sure that we kind of are on the same page. Whatever I'm telling, you are also understanding over there. Think about that. This is a virtual class, so we don't have the opportunity to interact one to one, and therefore I assume that and take it for granted that whatever I'm telling, it's been conveyed to you, right? In case if it is not, feel free to email me, WhatsApp me, whatever the way you can contact me, connect back to me and tell that, okay, this is the problem we couldn't understand, and then we can actually go back to this again. Okay, now I'm going to explain each one of this very slowly, so that we understand it. Very important. What are the features of a system? So when you say systems biology, then we understand that what is a complex system. The first two slides, we understood that what is the importance of the systems biologies. Why do you need the systems biology? We are also seeing the example of a few of the complex systems. We also understood and we articulated that why a simple system is not sufficient, because the biology which we have been doing so far has been reducing a complex system into simpler components. But then when you try to extrapolate it back to the original system, then often it fails. And therefore it is better to understand the whole complex systems, taking into consideration that everything is being joined together, connected together, dependent on each other. So it is an interdependent, interconnected, right, system, which you are actually seen in the earlier definitions. Okay. Then now we are coming to something very important, which is features of the systems, or what are the attributes of the systems. Okay. The first is components or the constituents of the system. Now these words actually sounds very similar when I say components of the systems and constituents of the systems. Now what are the difference between these two? What is the difference between components and constituents? Okay. When I say components of the systems, this means what all things makes up the system. That's the definition of components of the systems. What are the components of the systems? What all things makes this system, right? And when I say constituents of the systems means what is the fundamental plan on which the system is built up of. I'm going to give an example to make it very clear. So components means the different things that actually remains in the system, that exist on the system. That's a component. And what is the constituents? Constituents means the fundamental plan of the system, the fundamental way in which the system is built up of. I'll give an example. That's a cell phone. Every one of you have a cell phone, right? So if you see the cell phone and open it, we'll have hundreds of apps, right? You will have your Gmail, you will have your WhatsApps, you have your different apps which has been loaded in this particular phone. Those are the components of the system, right? But what is the constituents of the system? The constituent of the system is the motherboard of this particular mobile phone, because that is what actually makes it first in the first place. And once that is made, so many different things can live in this system. So that's the example. I'm going to give you another very good example. Say for instance, ocean. In a big vast ocean. When I say what are the components of the ocean? Well, the components of the oceans are the hundreds and thousands of fishes and animals and plants that actually inhabits that particular ocean, right? If you see an ocean, there will be millions and billions of species of fishes and other animals which has been surviving on that ocean. So those are the components of the systems. But when I say what is the constituent of this ocean, The constituent of this ocean is just water, H2O, right? That is what makes the whole ocean. That's the basic fundamental plan of the system. But once that forms the system, then many things can stay or leave on the system. Those things that leaves on the systems are known as the constituents, are known as the components of the system. So there's a difference between components of the systems and constituents. Again, I'm going to repeat it, that when we say components of the systems, it means the hundreds and thousands of the things that coexist on the system. And when I say constituents of the systems, means those things that makes the system. There's the difference between these two. Something like water makes the ocean. That is the constituents. But once the ocean has been made out of water, then millions of organisms and animals and plants can stay into that water in the ocean. That is the components of this ocean, right? So I think I had made it very clear to you what is the difference between these two. The next important definition is, what do you mean by interrelated components? Well, interrelated components means what is the relationship between these two, between the different things that actually stays or lives on the system. So that is the relationship between these two. I'll just give an example. Like in Japan, one of the things which is very common, which they kind of legally allow you once in a year, is to hunt whales. Whales are endangered animals, mammals, right? But in Japan, you have once in a year, for 7 days or 10 days, you could go and kill any whale. There is a whale hunting program that goes on over there. Whatever weird it may be, but that's what happens in Japan once in a year for a week time. Now think about it. When people actually did some experiments and then observations, and they found that when whales are getting killed in those 1 week, then there is the colorations of the color of the ocean in those regions turns very violet in color. Okay. Normally the ocean color is very blue, but if you see in those whale hunting periods when massive number of whales have been hunted, then the ocean color changes. It changes into a little violet in its nature. What is the reason for that? Well, when naturalists and when sea explorers try to go deep into it, when they dig further deeper into this, they found that normally the whales used to prey on a particular fish, and those fishes used to eat a particular planktons or sea animals, small sea vegetations, and those planktons produces a violet colored pigment. Okay, so whales used to eat a fish, those fish used to eat the planktons, and those planktons are going to produce a violet colored pigment, right? Now when the whales have been massively hunted, then there is a local suppression of the whale population in that region, and therefore those fishes which the whales used to hunt, their population increases, right? And as their population increases, they used to eat up those planktons more, and therefore there is a difference in the color, because normally what happens is that this small fish is going to eat the planktons, and the plankton actually produces the violet colored pigments. Now if the fishes are been surviving more because there is less of whales, then there will be more eating up of the planktons of those which are going to produce those violet colors, and then the color of the ocean in that region actually changes. So these are all interrelated with each other. See, hunting of the whales has to do with the change in the color of the ocean, because of the reason that they have been interconnected with each other. So there is an interrelationship between themselves, an interconnection between themselves. Again, interrelationships and interconnections are different. I told you in the previous slides that when we talk about interrelation, we talk about even binary things. When you say about interconnections, we talk about a very complex network of connections, right? The next important attributes of the systems is what you call as the boundary of the systems. What do you understand by the boundary of the systems? So whenever a system actually has been there, it operates, it exists up to a particular limit, and after that there is a beginning of another system. So every system actually operates within a boundary, like a human body. Think about it. That's up to where our boundary is. Beyond that, we don't have any control on ourselves, right? That's an environment. So boundary is actually the interface, or the boundary is actually the perimeter which the system actually occupies. Beyond that, the system is over and the next thing it begins with. Okay, that's the boundary. The next term what you use in systems biology is known as purpose of the systems. What do you mean by the purpose of the systems? Purpose of the systems means what is the motive of the systems? What is the motive of the complex behavior of the systems? Like for instance, if you have glycolysis which has been going on in an anaerobic condition, then what happens is that there is more formation of lactic acids on the pyruvate, right? Because the pyruvic acids cannot enter into the T cell cycles efficiently because there is less of oxygen. And as a result of this, the entire pyruvate gets diverted into forming lactic acids. So, the purpose of the system is that to produce lactic acids whenever there is an hypoxic conditions, whenever there is a decrease in oxygen. So that's the way in which the system actually gets rid of the pyruvate which is actually been formed because the pyruvate cannot enter into the T cell cycles. Okay. So that is the purpose of the system, that how the system actually copes up with the different conditions, with the different changing environmental conditions. Okay. And therefore we use the next word is that environment. Environment is anything which is out of the system. And therefore the boundary is very important over here because the boundary is the interface between the system and the environment. So think about this as a system, and anything outside that system is actually going to be environment, and boundary is actually going to be where the system ends and the environment actually begins with. And very related to that is interfaces. Interfaces means where the system ends and the environment actually begins with. And it need not necessarily to be that, it can be also within a complex systems where what are the different stratas or whatever the different hierarchies, what are the different interfaces. I'll just give an example. Think about an RNA which is actually been coming out from the nucleus. It comes out of the nucleus and then it actually interacts with the ribosomes, right? It sits on the ribosomes and then it actually encodes for the ribosomal protein synthesis. And once the protein is actually been synthesized, then that protein through the ribosomes enters into the endoplasmic reticulum, and from the endoplasmic reticulum it goes to the Golgi apparatus, and from the Golgi apparatus it eventually been thrown out of the cell or stays into the cytoplasm of the cell. That is the normal route in which the protein is actually synthesized right from an RNA. So at every stage we could see that there is an interface. So there is a nuclear interface from where the RNA actually exists, and then it actually begins with a ribosomal interface where the nascently synthesized RNA molecules gets integrated into those ribosomal subunits, and then it actually gets translated. And once it gets translated, that protein no longer remains in the ribosomes, it actually enters into the endoplasmic reticulum. So then we have the interface of the endoplasmic reticulum. Once endoplasmic reticulum, the protein actually gets glycosylated or change in the conformation, then it actually goes to the Golgi complex. There it enters into another complex systems where again there is substantial amount of modification by addition of the glucose residues of the sugar moieties, right? The protein gets glycosylated. So there is a Golgi interface, and from the Golgi, when the protein is actually been formed, then it actually gets exported out either of the cell, and then we have the membrane interface, or it stays within the cells. Then we have the cytoplasmic interface. So interfaces means the different layers where one system actually ends and another system begins with. So we have the nuclear interface, we have the endoplasmic reticulum interface, we have the ribosomal interfaces, we have the Golgi interface, we have the cell membrane interface. These are the different discontinuity within a particular complex systems where there is a demarcation in the functions. Okay, that's what is known as interface. Now next we are going to talk about input of the system. What does that input means? Input means what goes into the systems, what you put into the system. Very simple. Input means what goes into the system, what puts into the system. Output means what comes out of the systems, right? When you eat food, that is the input, the food as a material. But what it gives out, what comes out of using that food is the energy molecule. Suppose for instance ATP. So those are the outputs of the system. So input is the food and the energy is the ATP. Because the body as such cannot utilize the food unless and until it converts them into an energy currency, that is the ATPs in our case, right? And then we have something called the constraint of the systems. What does that mean? That means every system has its limit of operations, and beyond that the system breaks down. And simply you cannot use the systems to work for every conditions or any circumstances. So that is what is known as the constraint of the system, that the system is been constrained to function in a particular way, doing a particular responsibility. Beyond that responsibility, the systems cannot function and is going to break up. That is what is known as the constraint of the systems, the restriction of the systems, the boundary beyond which the system no longer functions as a system. And that is the scenario what happens in the cell death or apoptosis, where the entire system actually collapses, because those interfaces are actually been lost, and we don't have a very clear cut interface between the systems. Okay, so that is what is known as the constants of the systems. So again, going back very quickly to the features of the systems, we have the components and the constituents of the systems. I'll explain to you what are components, what are constituents. Components are the different things that actually exist into the system, and constituents are the raw materials that makes up the systems. Like ocean, water is the constituents of the systems, and in the oceans there are hundreds and thousands of the animals and the plants. Those are the components of the oceans, right? So constituents and the components. Interrelated components means when one actually affects the other. I give you the example of the whale, where hunting of the whale has a profound influence on the coloration of the ocean, because it actually alters the amount of the pigment which is actually produced by the sea plankton. And then we have talked about the boundary of the systems, where the system actually ends and then the surrounding begins with. And then we talked about the purpose of the systems, because every system is programmed or tuned to do a particular function. That's the philosophy of the systems. Like for instance, TCA cycle is going to produce reducing equivalents in the form of NADH and FADs, right? And which goes into the TCA cycle and electron transport chain, and then eventually it actually produces either 3 ATPs or 2 ATPs, depending upon whether it is an NAD or whether it is an FAD. So from the TCA cycles, the reducing equivalents in the form of NAD and FAD goes into the electron transport chain, produces ATP. So that is the purpose of the system. The system has been designed to function in a particular way to give a particular output, right? That is what is known as the purpose of the systems. Environment means anything which is out of the system is known as environment. The surrounding, which has been just around the system, is known as environment. Interfaces is actually the term which is actually used to denote how one system ends and another system actually begins with. So we have given an example of how the RNA actually gets translated into a protein, and eventually the protein gets secreted out of the cell. And we talked about what we call as the interfaces, where we have this nuclear interface, and we have the ribosomal interfaces, endoplasmic reticulum, Golgi interface, where every stage is a new system begins with and the old system ends, right? That's what is the interfaces. Input means what goes into the system's input, and output is what comes out of the system, that's output. And then we talked about constraints. Constraints means the limitations of the systems, the inefficiency of the systems to do everything, right? The systems cannot do everything, so there is an inability of that one. So within a particular limit, the system is going to work and operate, and after that the system is going to come to its constant where it actually cannot operate, and that is known as the constraints of the systems, or the limitations of the systems. So these are the features of the system. So if I give you an example, and if I tell you, tell me in an exam that what are the functions of the system, what are the features of the systems, you need to jot down all the different points and actually explain at least 1 or 2 lines, so that it actually been convinced to the reviewer or that to the examiner that you actually have understood this particular system's biology. Okay, these different terms, very important. Okay, this is a cartoon that is actually showing you the examples of a few complex systems, like for instance the human body, the fighter jets, a very complex microscope, a car, a complex machine like that. So why we are telling this as a very complex system? Because if you see any one of this system, there is not a single thing that actually makes it up, right? There are hundreds and thousands of the pieces that actually interact with each other to form the complex systems. Like for instance, if you take a fighter jet, if you open, even if you enter into any jetliners or any jet plane, then you will see that there are millions and billions of pieces which are interacting with themselves in a very complex way, and that actually makes the systems to fly and then do all these maneuvers and whatever it is. So that is actually an example of very complex systems. Very simple example: take the car take the car example of a car, right? In the car, if you see, there are so many different components that actually interacts for the car to be functional. We have the engines, we have the gears, we have the clutches, and then we have the suspension systems, we have the fuel systems, we have the air conditioning systems, we have the sound systems, we have the electrical systems, music systems, hundreds of systems that are all integrated into one platforms. So that is what is the examples of a complex system. So if I ask into the exam that give an example of the complex systems, then I'm going to talk about this and explain at least into 2 or 3 lines. Why do you say these are complex systems? So what is the criteria for this one to become a complex systems? Okay. And then we have to explain in the way exactly I told you, that how each one of them actually made up of so many different components, and each one of them actually interacts with each other in a very complex way. That is an example of a complex system. Okay. let's see the new slides. Okay, then this is the new slide which I call as the triangle of systems biology. What is triangle of systems biology? Means what are the different components of the systems biology? Look at these slides. So this is a very important slide that tells us that systems biology is actually been composed of three fundamentally more important components. One of them is known as the biological components, the questions which you are going to ask. The second is known as the computational components, the one which actually does the calculations and algorithms. And the third is the technological components, where the technological components actually gives an upper hand of how efficiently the systems can actually be probed. Think about it. If you don't have a PCR machine, you would not have been able to do a real-time PCR or a regular PCR, right? So the technological innovation is very important because that is going to help the computational systems, and together they are going to address the biological problem. So that's the triangle of systems biology, where it is actually an integration of three different components. Biological components, very important. A computational component is going to do some complex mathematics and arithmetics and draw some conclusions out of it. And then you have the technological components, which is actually the thing that actually gives the technological blueprint of the system, that how the system is going to work, how the system is going to operate. That's what is known as the technological component of the systems. Okay, so far we have any issues? I'm just going to see if any one of you has raised any points or not. Okay, let me check. Any doubts so far? Before we go into the next forum, okay, I assume that maybe you all of you have lots of doubts which is beyond the capacity of you possibly to write it down, or you don't have any doubts, which is very unusual. So again, feel free to get back to me whenever next time we meet, or any other way of communicating me, because this is a little difficult subject, so we need to pause and understand it. You cannot go it in a hurry, because then then you are going to mess up with the whole thing. So we are going to pause it and proceed further. Okay, wonderful. So let's proceed further and see what can be done. Okay. So I'm again pulling up the PPT which I'm supposed to show you. Okay, let the PPT to come. Okay, so before it comes, I could see the PPT, so let me explain to you that what are the overlapping things in systems biology. In other words, whenever we study systems biology, okay, what are the different subjects that actually enriches this particular domain of systems biology. So systems biology is a rather new field. I mean, 10 years before, people had not even heard of the word of systems biology. Trust me. It is just in the last 5 years or 7 years, people are talking about systems biology. So it's a very nascent field which is still rapidly developing, but people know the importance of it. It's so much important that you can answer a lot of complex questions using principles of systems biology. And there are a lot of things that actually goes into systems biology. We call as the different components of the systems biology. Remember, you're talking about components and constituents. The different components of the systems biology and the different constituents of the systems biology are different, right? Systems biology is actually essentially involves so many different things like bioinformatics, computational biology, okay, studying of the molecular components, writing algorithms, doing computer programming, taking large data sets and doing the data mining out of it. So all of this actually comes within the systems biology. So if you say systems biology is a very complex field, this actually involves so many different things that actually helps to navigate into systems biology. So this subject is actually been enriched by mathematicians, engineers, computer biologists, just hardcore biologists, and then anatomist, the microscopist, and so many different things who actually try to understand the subject in their own way and design the systems biology. Okay, that's very important. Now this is a slide which actually talks about the different things of the systems biology. Now this is something which we had already discussed, so I'm not going to spend too much time into it. This is another overview of the thing, which same thing which I actually explained to you a little back, that we could have different dimensions of the system. So whenever you talk about systems biology, again there is something that goes in the mind, that most of the time we possibly think that it's a very complex thing, so it needs huge complex organisms like humans or any other eukaryotic organisms. But that's not the case. When you talk about systems biology, it could be a small complex systems, it could be a large complex systems. So we could have systems biology which can be studied for a complex systems like human beings, or systems biology which can be studied for a very simple systems like the unicellular bacteria. So anything can be actually be under the domain of systems biology, depending upon what are the things, whatever you are going to look. So over here I'm going to be a little philosophical and tell that systems biology is actually a mindset. Systems biology is actually a mindset or an approach in which you address a problem. Even more than it as a legitimate subject, it is an approach how you are going to tackle a problem. Say for instance, if I tell you this is the molecule which has been possibly causing cancer, or this gene is actually been mutated in cancer. Now go and find out all about this particular gene. So there are different ways and different approaches of doing that one, right? The different approaches will be, suppose for instance, in a systems biology approach, what you're going to do is that you're going to find out the gene. We're going to see the chromosomal location of the genes. We're going to see how this particular gene is going to interact with the neighboring genes, whether they forms a particular operant kind of a network or not. Then you see what is the protein product of the genes. What is the nature of the protein product of the gene? What is the biological function of those protein products of the gene? So you could see that we are going to address hundreds and thousands of questions. Doesn't matter whether it is a complex systems or whether it is a simpler systems. So the whole take-home message is from this slide is that it is irrespective of the nature of the systems, how complex it could be or how simple it could be, you could still apply the principles of the systems biology to address the different fundamental questions. So it is more of an approach from your end. It's more of a mindset from our end that how we are going to tackle the problem. We are going to tackle the problem as if the problem is in isolation, like a reductionist point of view, or as if the entire system is been complex and connected with each other, and that is from a systems biology point of view. So that is what has been actually, everything has been actually there in this slide, is that you can study a large complex systems or you can study a small system. Okay. So the next is that why study systems biology? Okay. Why study systems biology? Because of the fact that, let the slide to come. So there's a small animated slide which actually talks about why you study systems biology. This is something which I had already described to you. This is with the help of a cartoon, trying to make you little more convincing to whatever I told you. Okay. Now, if we study what is time t is equal to 0, this is perhaps the time when people actually started studying science as an experimental branch or as a theoretical science, whatever it is. The time t is equal to 0 is possibly the time when you started studying science in a very disciplined manner. And time t is equal to today's time when you actually studying biology right now. That is what the history of the sciences, right? So in the history of the science, majority of the time we have actually studied biology as a reductionist point of view. That's what I was actually telling you again and again, that we take a very complex system and break them and tear them apart into smaller components and study each of those smaller components, right? So give an example is that, say for instance, this particular examples where we have these organisms, right? A very complex organisms. Think about it. So let the slide to come again. So this is a very complex organisms, and what we are trying to do is that we took their organisms and then harvested the organs from their organisms, right? And from the organs we had actually harvested the tissues, and from the tissues we have harvested the cells, and those cells which we have harvested, we grow them into tissue culture flasks, right? Any one of you who has been actually accustomed in doing cell culture or tissue culture, possibly you are going to learn it. If you have not done it, you are going to grow them into small petri dishes over here. This is the small petri dish, a small flask where you go and where you can isolate the cells and tissues and actually grow on them, right? And then we set up an experiment to find out that a particular hypothesis, and then eventually we have the data. And what we do is that with that data, we try to correlate back to the organisms. Think about it. This is the data, and then from the data, we try to go back to the organism. Or in other words, we take a very complex system, and for the ease of our understanding, we break that complex system, we tear apart that complex system into smaller components, cells, tissues, grow those cells, tissues into petri dishes, and then study the biology of that, and then try to correlate that data which you obtained from cell and tissues back to the complex organisms. This is the what you call as the reductionist point of approach, and most of the time this is the conventional point of approach where most of us actually do biology in this way, right? Now the problem is that when we try to correlate a very complex system, break it apart and bring it to smaller components, most of the time you cannot stitch back those smaller components to the complex system again. Or in other words, majority of the time it is an irreversible process. So if you take, say for instance, let me give you an example, that if we take, say, a cancer tissues, say for instance pancreatic cancer, one of the most common pancreatic cancer is PDAC, right, pancreatic ductal adenocarcinoma. Or if you take, say for instance, a bladder cancer, right, majority of the bladder cancers are actually transitional cell carcinomas. So you take that whole cancer tissue and then you break those cancer tissues into cells and then isolate the cells and do some fancy experiments on the cells, and then from those experimental data, if you try to correlate back to the original cancer, most of the time you will see the correlation is extremely poor because of the fact that you have destroyed a complex system into smaller components, and that smaller components cannot now represent that complex systems because that smaller components is too simplistic that it can actually address the complex systems altogether, and therefore we lose certain informations, even though this is much more easy for us to do rather than actually working on the complex organisms per se. But at the same time, it is also very hard for us to correlate back to the original system once we break them into smaller components. So that is something which I just wanted to put a caution to you, that whenever you are actually studying conventional biology, think about this kind of things which is happening every day and night. Whenever we set up an experiment, we try to have a complex system broken down into smaller components, and then we try to correlate back to the complex system. It never works in that way, right? It is very hard because of the systems are most of the time it is actually irreversible. I'm going to bring the next slide that actually talks about what is the dilemma of the biologist. Okay. This slide has come. So over here there is an issue that if you take a complex systems and break them into smaller components, why we cannot correlate always? Because those data which you obtain from that smaller components are not being correlated back to the complex because of the reason that this is an example of a non-linear system. Now I had actually introduced a very important term that is known as non-linearity. What does that mean? It means that 1 plus 1 plus 1 does not always adds up to 3. It could mean anything. So in systems biology approach, what we problem is that you cannot tear apart the systems and study them separately and then join them together. But this process of studying them separately and joining them together is known as additivity. That is, you can add on the components informations together to form one composite things. This additivity is not been possible in the case of a complex system. So the moment you tear them apart, you really don't have any clue whether their effects can be added in a linear fashion. And that is the reason why we could not extrapolate every single thing, whatever you obtain from in complex systems, by breaking them into simpler components. I'll give another example of this one, is that if you take in tumor tissue, suppose you go to the, say, breast cancer, right? You go to the clinician, the breast cancer surgeon, and he or she will actually give you a tissue, right? And then you took the cancer tissue from that particular sample, and then in order to compare that cancer tissue, he also asks the surgeon is that, can you kindly give me a normal tissue? So what the surgeon will do is that with his or her best educated guess, they are going to go a little further from the core of the cancer and is going to give you so-called the normal tissues and give it back to you. Okay, this is far away from the cancer. Now this is a normal appearing tissue to my eyes and possibly to an histologist, and therefore why don't you take these as a normal sample, right? So this is known as the matched controls, because we took the normal or the control right from the same patient, the patient who has the cancers, right? So that is known as the matched control or the normal controls. Now, for the surgeon or for a person who's actually been looking in a macroscopic way, in a very superficial way, this is possibly okay, because still the cancer features has not come into those so-called normal tissues, and therefore we say that they are pretty normal. But if you do a molecular study, you will find that those tissues, so even they are far away from the core of the cancers, most of the times are actually been primed by the cytokines which has been released from the cancers, and therefore we may not have an absolutely normal cells which has been there surrounding a particular cancer. It is just not possible. So we call them as a cancer field effect, where a particular cancer can actually influence its own surrounding by the release of different cytokines and other factors, which kind of primes that particular environment, so that this particular environment is been actually has an effect. So what is the problem with this kind of approach is that whenever now you are trying to compare with the data, so with this so-called control, you will see that we really don't have a normal behavior of the control. We have the cancer field effect. So even though the surgeon may not see visually the changes in those surrounding control tissues, but actually there are molecular changes, and therefore they cannot serve as a perfectly good control. So if you suppose do a microarray experiments, or if you do a next generation sequencing experiments with that particular cancer core and so-called a normal core which a surgeon has given you far away from that, even then you may not see a perfectly normal behavior of that particular thing, because of something called the cancer field effect, where this particular cancer tissue, by the release of these different cytokines and other small molecules, has primed its entire surrounding, so that the surrounding is no longer just like a normal tissue, but it has been little bit tuned and altered because of this cancer presence of the cancers. And that is very important for us to take into considerations. So these are the things which is possibly ignored when you study when you don't study systems biology, but look into a classical way. But the problem is that in systems biology, good thing is that in systems biology we kind of address all of these problems and take into considerations of the entire surrounding, not just the tissue, but the entire surrounding to that of the tissues. And that is what is actually been explained over here in the previous slides, that it is very hard for us to break down a complex systems into smaller components and do a biology of the smaller components and go back to the complex systems, because of the reason that there will be so many different effects on influence by the environment and the surroundings that you may not have a real life scenario that it can be always correlated back. Okay, so that is something what I was talking about. Now over here in this slide, it is been said that this is not a unique problem in biology. So it's not that biologists are actually been complaining about these problems. Physicists has always encountered this problem way back, that if you have a large system and if you try to reduce it and bring it back to a smaller tiny system, then many of the times they don't correlate, right? Because of the reason that they have been actually joined, or they have been actually related with each other, not by simple way but in a very complex way. Now here actually comes what you call as the linear relationships and the non-linear relationships. So this is possibly the one which I am going to talk, and then you are going to stop the class, because after that it will be a little more overdose for you. And what I will do rather is that I am going to summarize the entire class, so that it becomes a little easier for us to understand whatever we had actually discussed so far. So this is the problem. What is the reason? Now many of you may be wondering that what is the reason why we cannot do that one. Okay, now I'll give you before going into this particular slide, I'll give you an example of this one. What is the example? This actually really happened with me when I was actually doing a research as a PhD fellow long back. What happened is that we had a lab colleague. She has to perform an experiment where in one of the cell lines she is going to stimulate with a cytokine number A, and with the other cell line she is going to stimulate with the cytokine number B. Actually, I'll tell you the name of the cytokine. She was stimulating one of the plates of the cells, one of the plates of the cancer cells, with a cytokine that is known as interleukin 1 beta, and she's stimulating the other petri dishes, the other cancer cells, with another cytokine that is known as tumor necrosis factors, or TNF alpha. Okay. Now, her motto of this particular study is that what are the changes in this cancer cell is been brought about by the cytokines IL-1 beta and TNF alpha. Okay, she's going to study that. Now, what she did is that she has also to address a problem that if we add both IL-1 beta and TNF alpha together, what is going to be the effect on those cancer cells? So we have three different experiments. One is that just add them single, and next is that add them together, and then see whether there is an additive effect or whether there is a non-additive effect. Because we know that these are all pro-inflammatory cytokines, IL-1 beta and TNF alpha. So if we add each one of them separately, what is the effect, and what is going to be the effect if we are going to add all of them together in one particular cell? Okay. Now she made a mistake is that she has forgotten to add all the two together. That particular experiment she forgotten. So she added just IL-1 beta. She just added just TNF alpha, and then she sent the samples for doing a microarray analysis. Microarray analysis, for the people who are not aware of, this is a technique by which we could find out the gene expression or gene expression signatures in a particular cell type or whatever it is. Now, after doing these experiments, she suddenly recollected that, I forgot to add both of these two cytokines together. So why don't I do something that I get the IL-1 beta data, I get the TNF alpha data, and then I'm going to combine them together. And if I'm going to combine them together, these two data, mix them together and or add them together, then I'm going to possibly get the data which I would have otherwise got if I've added the two cytokines together. In other words, if we add one cytokine, if we add another cytokine, get the individual data and add them, that whether that is same like adding the two cytokines together and then getting the data. In other words, can we digitally add them even though we actually have not performed the experiments in adding them together because we forgot it or whatever the reason it is, right? Now when she did this experiment, she found that when you add the data separately, you are not producing a good picture. Or in other words, it doesn't matter whether you actually have some very good data of individual IL-1 beta or TNF alpha, but once the moment you have not added them together, you cannot recapitulate that. You cannot capture that information by combining these two data separately. And this happens only when the system actually is a complex system and actually behaves into what you call as the non-linear system. So in the case of non-linear systems, the simple rules of additions or the simple additivity laws or rules does not hold good. Now this is what is actually been explained in this particular graph. It actually talks about a linear systems. Linear systems are the systems where output is proportional to input. Or in other words, if you give 10 nanograms of a cytokine, you are going to get this much input. If you are going to add 20 nanograms of a cytokine, you are going to get double the effect. If you are going to add 40 nanograms of the cytokines, you are going to get 4 times of the effect. In other words, these kind of things that actually happens is what you call as a linear relationships. Okay. And the other example of the linear relationship is that what I had actually explained just now, that if you add one cytokine and get some data, you add another cytokine, get another data. Now you combine these two data. Does it really produces the effect of adding as if we are actually adding two cytokines together? No. The reason is that the biological systems are not linear. So before that, what is not linear, we're going to first see what is a linear system. So linear system is a system where the output is proportional to the input. Or in other words, how much you are going to get the output, that much is actually dependent upon the inputs. So if you add more and more of inputs, then you get more and more outputs. We are seeing in many biological systems where even if you add lots and lots of substrates, the enzyme kinetics does not become much more robust, right? The enzyme actually reaches to a state of saturations. And this is because of the fact that this is actually not a linear relationship, this is a non-linear relationship where the output and inputs are not proportional to each other. They may be proportional to some extent, but they are not completely proportional to each other. If it happens that the output is proportional to the inputs, if you add 10 nanograms of the cytokines, you get this response. If you add 20 nanograms of the cytokines, you're going to get double the response. If you get 30 nanograms of the cytokines, you get 3 times of the response. If this kind of condition happens, then you call them that is a linear relationships. When it does not happen, then it's no longer a linear relationship, then it is a complex relationship, or it's a non-linear relationship, which we are going to discuss just in the next slides. So this is a graph that actually tells you the equation of a straight line, y is equal to mx plus c, and in this straight line equation you could see that if we know the values of one of the variable, either y or x, you could always derive the value of the next variable. So this is very simple. In other words, the one particular variable is going to decide the fate of the other variable. Now that is the epitome of a classical biology. But in systems biology, it doesn't happen that simply, because the output and inputs are not always proportional to each other, because of the reason that there is so many different complex things that is happening, so the output and input does not matches. And that is the reason why we cannot have an additive law or additive rule, whatever we have in the classical biochemistry, but we have that rule in the case of systems biology, right? So this is about talking about a linear systems. So a linear systems can be followed by simple laws of additivity, or in other words, you could actually add the two components together to form as if you got a combined effect. Now the linear systems can actually be represented into what you call as the two-dimensional space, which is known as the Euclidean space. So this is how a linear systems can actually be represented on this. Okay, and the linear systems are linked together by simple linear transformations of additivity and homogeneities. So this is the way in which the linear systems can actually be done. We can actually add them together as if they are actually been stitched together. But in the case of complex systems, you cannot stitch. Say for instance, I give another example like TGF beta, which is transforming growth factor beta, which is a very important cytokine. Now if you add TGF beta, if you keep on adding TGF beta, you get a particular response. But if you keep on adding TGF beta for on and on, then you'll see that after some time, the entire response goes down in an opposite direction. In other words, there could be an increase in the expression of a certain molecules by the help of TGF beta, but if you keep on adding TGF beta, it doesn't goes on increasing. Rather, now it is going to have a dip. And this is what is known as the biphasic response, where we have two phases, a climbing phase and a declining phase. And this is mostly being possible because of the system, because of this nature of the systems. Okay. So in that case of TGF beta example, why I said, because if you keep on adding TGF beta, you may not have a linear increase. Even though you are adding TGF beta, after some time you will see that there is no further increase, rather there is actually a decrease in that trend, and that is actually very important. So this is the examples of a linear systems where things are actually been added in a linear way, where the principles of additivity and homogeneity apply. So the principle of adding the features of the systems is possible, but this has not been possible in a complex systems, which is the example I given you just now about the TGF beta examples, where you cannot keep on adding TGF beta and get more and more response. After sometimes you get in, or you reach to a point of saturations. Now with this I am going to end this particular slide, and I am going to summarize the today's class. Even though we have not completed yet, we are going to complete the rest of the things in tomorrow's class, but I am just going to summarize whatever we had actually studied so far. We had actually studied what is systems biology, and we had also defined systems biology in the terms of a few attributes, right? We have first defined it that it is an interconnected, interrelated, interdependent, interacting systems. Remember that? The first definition, very important, that it is a collection of the things which are interdependent, which are interacting with each others, which are interconnected with each others, and therefore this whole forms one complex systems, not individually. We have also studied how the biologists used to study biology in a reductionist point of view, where they take a complex system, break down into simpler components, study the simpler components, and try to extrapolate back to the systems. Many of the time that actually failed very badly, because of the reason that simple laws of extrapolating cannot hold true if the relationship is not linear, rather it is a non-linear. And then we had seen what is linear, what is non-linear. Linear is when there is a predictability, or when you can predict some things. Say for instance, if you can predict that how the system is going to happen at every time point in the future, you have a linear relationships. But in a non-linear relationship, there are so many different variables that steers the systems, and therefore just having a few parameters cannot let you to project and cannot let you to forecast the systems very easily. And that is an example of a complex systems. So linear systems are actually has a very high predictability value. They can be explained with the help of a simple equation, y is equal to mx plus c, and they are actually much easier to handle. On the other hand, complex systems are the true systems, the biological systems. They are non-linear because of the reason that they cannot be joined together by the rules of additions or additivity, and therefore they are not, or they cannot be actually studied by the reductionist point of view, where you take a complex system and break it down and try to extrapolate it. Okay, and fortunately, unfortunately, whatever the way you can think of, our biological systems are all very complex systems which behaves in a non-linear fashion. It doesn't behave in a linear fashion, but in a non-linear fashion. And therefore, that is the problem, or that is the fallout of this entire thing, is that we cannot have the simple laws of additivity in our complex systems like our biological systems. Okay. So with this, we kind of end today's class. We are going to continue from here for tomorrow's class. And if you have any doubt, just jot it down on a piece of paper, and then before we begin the tomorrow's class, just explain me. Just come and talk to me, or just write an email over here, or put it into the chat notes, and bombard me with your questions. No, not an issue, but just try to speak out, so that I get a little bit idea that you're actually following this class, because it is more mathematical, it's more analytical, so it might be difficult for few of you to follow the intricate details of it. But bear me, if you actually study and go through the slides at least 7-8 times, very quickly, if you brush through it, you will become very accustomed with everything of whatever has been actually mentioned in the slides. It's very logical and very convincing, and therefore it is actually not that hard what it actually appears to be in the first class. So with this, I actually end over here, and then actually see you all tomorrow with the concluding portion of these slides. Okay, so thank you, have a nice day, and stay safe. So thanks a lot. हो गया हमारा.
एंड कर दूं सर?
हाँ, सिंपल.